Good morning. My name is Kanahan, Assistant Professor, Department of Pharmaceutical Analysis and Chemistry. Today, my topic is factor affecting ion exchange chromatography. So, we are using ion for the chromatography. Chromatography means a separation technique using to separation of multiple ions or molecules, atoms, whatever it is. So, what are the factors involved in affecting the ion exchange separation? First one is nature and properties of ion exchange resins. So, what are the nature or the ions or cross linking and swelling? It is most important for factor affecting. So, second one is the most cross linking occur. They are more rigid, but swelling is less. Okay, then swell less. The separation of ions, different sizes difficult. Okay, first one is nature of properties of ion exchange resin. Second one is nature of exchanging ions. So what are the nature in exchanging ions means? First one is valency of ions and size of ions. Some ions are large molecule size, some ions are smaller size. It leads to affect the ion exchange chromatography. Then polarizability and concentration of the solution and concentration of charge of ions. So concentration of solutions are liquid or percentage, and concentration of charge of ions means passive ions is more than negative ions, and pH of the mobile phase, pH of we are changing the mobile phase that leads to affecting the ion exchange chromatography and ionic strength and mobile phase modifiers and temperature. It lead to increasing temperature or decreasing the temperature leads to affecting the ion exchange chromatography. And what are the principle involved in ion exchange chromatography means? First one is equilibration and sample application and wash and elution and regeneration. Okay, first one is equilibration means process of its equilibration is the equilibration step. The ion exchange chromatography is equilibration. The first step is the equilibration of stationary space and desired to start conditions we ready to start. When equilibrium is reached, all stationary phase charged group are associated with exchangeable counterions such as chloride and sodium. So by the blue counterions is means counterions, and red ions means stationary phase is the group of ion is the red ions are stationary phase, and negative ions means counter ions is the chart of absorption and time of the volume. And second phase is the ion exchange chromatography, the sample application and wash. First one is equilibration, and second step is sample application. We are applying the sample. So second step is sample application and wash. The goal is the step to bind the target molecules. Okay, and finally wash out of the unbound material. Only bound only we are taken, and sample buffer should have the same pH and ionic strength and starting buffer in order to bind the appropriately charged protein. Okay, so these are the chart of the absorption. And third one is elution. So second step is completed, and third step, in the third step the elution biomolecules are elution from exchanger by a change in a buffer composition. A common way is to increase the ionic strength with sodium chloride, another simple sort, in order to the bound protein dissolve the bound protein. The protein are dissolved relative to the number of charged group in their surface. Okay, so these are the surface number of the protein ions or charged group present in the surface. These are the elution third step. And finally, these are the detector signal. So we are getting this signal. What are the molecules are we are gathering? So first one is aspartic acid and threonine and serine, glutamic acid and proline and glycine and alanine and cysteine, valine, methionine and isoleucine and leucine and threonine and phenylalanine and histidine and lysine and ammonia based upon the charge and charge of the molecule. Okay, so what are the applications involved in the ion exchange chromatography means? First one is softening and demineralization of water. Okay, that we are using the ion exchange chromatography for extraction of enzymes from tissues. So taking the extraction of multiple enzymes from multiple tissues from animals or plants. For humans, whatever it is, the purification of solution free from ionic impurities. Okay, so remove the impurities from the multiple solutions and enzymes or molecules or atoms, whatever it is. So separation of inorganic ions. So inorganic means not carbon containing compounds. Organic means carbon containing compounds. And separation of sugars and amino acids and protein. So ion exchange column in HPLC. Okay. Thank you. Thank you.